Derek Woodfield, General Manager for Grasslands Innovation. I've been involved in plant breeding and biotechnology research for more than 40 years, and I was a witness for the Royal Commission on Genetic Modification in 2000. Grasslands Innovation is an incorporated joint venture between PGG Wrightson Seeds and AgResearch. We currently invest more than $9 million annually in developing proprietary forages for pasture-based farming systems. We've been developing genetically modified forages in New Zealand, but testing these in Australia and USA because of the clear regulatory framework and clear pathway to market in these countries. We support the aims of the new Gene Technologies Bill to better align New Zealand with those in place in Australia and many other countries. In particular, we support the tiered science-based framework to assess the risk of new gene technologies and the inclusion of controls and risk management proportional to the risk. The current regulatory framework, as others have said, is outdated, overly cautious, and unworkable for introducing innovations into New Zealand farming. Despite the main findings of the Royal Commission stating that New Zealand should proceed with caution, the outcome has actually been an extremely cautious approach, with minimal progress as evidenced by the very few field tests of GMOs over the past 24 years. New Zealand farmers have not seen the benefits of new gene technologies and are increasingly at a competitive disadvantage compared to Australia, North America and South America. No genetically modified products are available to New Zealand farmers, contrasting sharply with the 93 foods listed from GM plants that are sold in New Zealand on the FSANZ website. We can eat them, but we can't produce them. Gene technologies are important tools in the plant breeding toolbox as we seek to improve the main plants upon which New Zealand's agriculture is based. We are actively investing in several GM products, including high condensed tannin white clover, that offer considerable potential to reduce greenhouse gas emissions, as well as improving animal health and productivity. There are no other methods by which these products could be produced. In the case of the high condensed tannin, outdoor trials are currently underway in Australia under licence from the Australian Office for the Gene Technology Regulator. We've also previously grown that in the States. The hurdles and significantly higher cost in gaining EPA approval for outdoor field trials in New Zealand has been a major factor in us going offshore to do these trials. Outdoor field trials in New Zealand will be an important step for each of these programs to demonstrate at scale their ability under New Zealand conditions to deliver the benefits that we believe that we've already shown in containment and in overseas field trials. These new laws, if enacted in New Zealand, would lead to us applying for field trials in New Zealand as soon as possible. We support the intent of the bill, the purpose and objectives of the bill to enable the safe use of gene technologies by managing risk, the intent of establishing a more enabling and modern regulatory regime for gene technologies and GMOs, the objective of having a nationally consistent approach to the regulation of gene technologies by removing local authorities' ability to restrict its use, and the formation of an independent single decision maker, acknowledging that this approach has worked well in Australia for the last 20 years. It increases the efficiency and reduces process time, and provides for clear accountability. Some considerations on the bill are listed in our submission, but include ensuring that the four-stage risk tier framework, that it's clear what fits into each of those and how that assignment will be made. We also would like to see in the establishment of the Technical Advisory Committee that committee includes people with practical knowledge in the primary sector and indeed in the plant breeding expertise. And lastly, we want to make sure that the terminology is clarified in the bill to ensure that it doesn't unintentionally include processes that are already naturally part of that process, including mutagenesis, which there are literally hundreds of cultivars already used out there. So in summary, I believe the bill is essential for New Zealand to reduce our environmental footprint to improve animal welfare outcomes and to ensure that we remain internationally competitive as a plant-based export economy. Thank you.
Thank you. Dave?
Thank you. You agree with your colleague from Ag Research that over the course of years and decades, it's not practical to consider that we could stop the release of genetically engineered grass from spreading?
I do. I think there's a lot of publications on coexistence. There will be gene flow, and so then it's how do you manage that?
So you manage the coexistence through allowing a permissible amount of contamination in those producers who don't want to have GE, but doesn't that just mean that you're actually sacrificing the true GMO-free status of the non-GE producer?
I think we have to measure that and assess it. I don't have data. I know for the white clovers that we've looked at, most of that spread is within 24 metres of the source, so it's quite contained. But there will be gene flow.
Excellent, thank you. Just two quick ones. So really to kind of follow up, you have a fairly good understanding of how pollen flows for grasses and clover. Is that what you're saying? Yes, excellent. You also talked about there's hundreds of examples of grasses. Is it grasses that you're referring to that have been bred through mutagenic processes?
In our case, probably the clearest example is our forage brassicas, which were developed using chemical mutagenesis. They've been out on New Zealand farms for approximately 20 years now, being actively used. But there are many examples worldwide. There's over 5,000 varieties that are listed on websites with chemical mutagenesis and other forms of mutagen.
Would you say that we have poor understanding of the outcomes from some of those processes that we would have from something, say, CRISPR-Cas9 processes?
With every one of those plants that have been released from mutagenesis, they have gone through exactly the same process that we would have to go through. We test animal safety of those products. We test their environmental resilience and fit. So they go through a very thorough process before they're released.
Do we need to label that?
I don't think so. I think the outcomes of those have been consistent with conventional products in every sense.
What are the risks with producing a novel grass species through genetic modification once it has been established and perhaps spread on us discovering something that we didn't notice early on?
That's a good question. Again, I think with every one of those plants, we take them through the same process. In terms of our grasses, they all go through an animal safety trial because they have a novel endophyte almost invariably in them. So they go through animal product testing for anything from 1 to 3 years. They also go through a range of environmental testing at up to 8 sites for 3 to 4 years. Right. So I think we do a good job of testing them to ensure that there are no unintended consequences.
Because you can create novel toxins potentially in a plant species through genetic engineering.
Yeah, I haven't seen any of those in terms of unintended or novel compounds. We do test we do a forage quality analysis on all of our products for those. I haven't seen any at this stage.
All right. Thank you, Derek. Thank you very much for joining us.
Just got to unmute myself there. Helicopter landing at Wellington Hospital, hence why I muted. So because the noise was travelling into the stream.
Yes, straight off. Did you notice how resistant he was to not only regulation but labeling, and how he got his 5 cents worth in about opposing the right of local councils to put in place any additional requirements? So obviously Grasslands Technology Australia, whose parent company is Ag Research, would like to undertake either outdoor experiments or releases, for example, in Auckland or Northland or Hawke's Bay, and completely disregard the wishes of local farmers and communities and councils. And of course, with the lack of civil liability, then any problems caused by the spread of the pollen or vegetative material or seed of these controversial GE grasses would fall on others to carry the can for the cleanup.
Yeah.
And,
that point about coexistence was really interesting. It was a tacit acknowledgement that we will have genetic material going between, basically the whole country, really,
Yeah.
And there was no kind of hiding away from it. Yeah,
And
more-
excellent questions from the Green Party MP, Steve Abel. And I've put in the chat a article by the investigative reporter David Williams. Grass
Yep, yeah.
isn't greener
Mm.
for GM trial in Australia. So the gentleman presenting obviously did not mention that earlier, was it last year? Yeah, 2 years ago, that they withdrew their application for the limited and controlled release of its GM grass in Australia. And essentially, they were not able to meet the standards required for a field trial in Australia. Sustainability Council Projects Director Stephanie Howard says AgResearch hasn't been able to get trials off the ground on either side of the Tasman, so it can't just be the New Zealand law that is the problem. And then Professor Jack Heinemann makes comment as well. So highly dodgy. I mean,
Yeah.
got to say.
We're going to have to wrap up because I've got 2 panelists waiting to come in. So,
Understood.
yep. Thanks for joining us for this hour. Brilliant meeting you both again.
Thank you, Linda.
Lovely to
Really
meet
appreciate
you.
That's all right. And yeah, we'll kick off for the next hour.
Thanks for watching. Make sure you're subscribed to our Facebook, YouTube, Twitch, Instagram, TikTok.